Okay, so we move on to the second step of cellular respiration after finishing glycolysis, which is the link reaction. You can see in the diagram on the left hand side of my screen that I've drawn a huge cell showing cytoplasm and also showing that inside the cytoplasm glycolysis ended up with production of two pyruvate molecules. Okay, we started with one glucose molecules and we end up with two pyruvate molecules, each containing a 3-carbon compound. Achha, now you can see that what is happening to this pyruvate. This pyruvate is entering into its structure. Yes, Abdullah.
Yeah, miss. So what, like when I did my reading, what I understood was कि glycolysis से जो pyruvate बनता है, then by active transport, ये cytoplasm से it moves into the matrix. Matrix में then it is decarboxylated and dehydrogenated, and then it is combined with coenzyme A to give acetyl coenzyme A. उसके बाद मैं confused.
Okay, fine. So this much is easy. So this means link reaction is the shortest reaction that takes place inside the matrix of the mitochondria. Essentially, what are the points that an A2 student needs to remember? Point number one, he needs to remember that the way by means of pyruvate enters the mitochondrion is through active transport. So energy kharch karni padegi, right? Or second point, what happens to pyruvate over here? It is decarboxylated, yani uska carbon dioxide nikala jayega wahan se, and it is dehydrogenated also. Hydrogen is also removed. As per the वो कि hydrogen जब remove होता है, ये दोनों चीजें हो जाएंगी, and then after that, this compound which is pyruvate will be now combined with coenzyme A to give another compound which is acetyl or acetyl coenzyme. This is known as a link reaction. इसको इन्होंने summarize इस तरह किया हुआ है. दोबारा से यहाँ पे ND का रोल आ जाएगा because wherever you see dehydrogenase which is removing hydrogens, तो NAD वहाँ पे जरूर आएगा उन hydrogens को carry करने के लिए. So the reaction can be summarized as pyruvate plus coenzyme A plus NAD gives rise to acetyl coenzyme A plus carbon dioxide plus reduced NAD. ये बात समझ आ गई कि NAD reduced NAD कैसे बना?
Yeah.
Right?
So wait.
Why? Because
ये जो pyruvate उससे combine होता है coenzyme A and NAD से.
हाँ. Summary है, ये summary है of the entire thing.
Yeah, but जब the NAD is reduced, how? Because coenzyme A जब pyruvate से बनता है तो acetyl coenzyme A बन गया, and then carbon dioxide बनता है, and then NAD reduced NAD बनता है because hydrogen is removed, right? And then वो NAD के साथ combine हो जाता है. Okay, sorry.
Exactly. ठीक है? अच्छा. So this is about it. This is the link reaction. Where does it take place? Yes, Abdullah?
In the matrix.
In the matrix of the mitochondria. ठीक है. सही. अच्छा, फिर आपको ये बताते हैं. This is an additional information के although we are doing, जब हम cellular respiration अभी पढ़ रहे हैं ना, तो हम सिर्फ ग्लूकोज की ऑक्सीडेशन का पार्ट पे पढ़ रहे हैं. But remember कि when we are starving, जब हम बहुत लंबी देर तक स्टार्व करते हैं तो हमारे फैट्स भी क्या होते हैं? They are broken down, isn't it? Fats burn होते हैं, especially exercise वगैरह में. So fatty acids from fat metabolism may also be used to produce acetyl coenzyme A. And fatty acids are broken down in the mitochondrion in a cycle of reactions in which each turn of the cycle shortens the fatty acid chain by 2 carbon acetyl unit. ये इसको बस इस तरह याद रख लो कि it's not only glucose जिसकी oxidation हमारी body में हो रही है. Fatty acid oxidation also takes place. तो जब fatty acid oxidation होती है तो at a certain point in time उसमें भी acetyl coenzyme A बनता है. Detail में हम इसके नहीं जाएंगे because that's not a part of your syllabus. Just to give you this much information is enough. So link reaction में I hope there are no questions. No?
Nope. All
Let's
good.
move on. अच्छा. All good. So ठीक है. ये तो उसकी summary थी. And let's move to now the Krebs cycle. Again, recapping, glycolysis happening in cytoplasm, link reaction and Krebs cycle happening in the matrix, और जो चौथा वाला इसका phase था oxidative phosphorylation that takes place on the crystal. But we still have to do Krebs cycle, so let's look at it. ये फटाफट आपको link reaction की एक summary नजर आ रही है, ठीक है? Now let me ask you questions. How many ATP molecules did I use in link reaction?
2.
None.
Wait, in link reaction? Excellent. Sorry. Wait, link reaction, man. You used
None.
pyruvate molecule. So wait. Yeah, there's no ATP. Sorry. Okay.
Acha. Say, how many ATP molecules did I make in link reaction?
None.
None. Okay. So this means link reaction is very unique. Neither you need any ATP to invest into the link reaction, nor you get any ATP from it. Right? अच्छा ठीक है अब हम आगे चलते हैं. This is the Krebs cycle.
Yeah.
Krebs cycle में क्या हो रहा है कि Krebs cycle में ये जो acetyl बना था ना 2 carbon containing compound था. देखो लिंक रिएक्शन स्टार्ट होता है 3 कार्बन से खत्म होता है 2 कार्बन पे और इस 2 कार्बन पे एक कोएंजाइम A लग जाता है and then 2 carbon when combines with coenzyme A it forms acetyl CoA. ठीक है. अब इस acetyl CoA के साथ क्या हो रहा है? Acetyl CoA will bind to a compound known as oxaloacetate, which is 4 carbon containing, to convert it into citrate. ठीक है. 4 plus 2 is equal to 6. Do you get it?
I get it.
ठीक है. अच्छा अब हम चलते हैं स्टेप बाय स्टेप. ये 6 कार्बन है. अब अचानक से 6 कार्बन का क्या रिमूव हो रहा है? What is being removed?
CO2. So it's
CO2.
decarboxylated.
Decarboxylated. And it's also dehydrogenated, isn't it?
Yes.
Yes. अच्छा, so I'm
Then it's
noting down.
decarboxylated. Then it's decarboxylated again. Then it's dehydrogenated again.
हाँ ठीक है ठीक है. So अच्छा decarboxylation number 1, decarboxylation number 2, dehydrogenation number 1, dehydrogenation number 3. अच्छा ये करते 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 हम 4 कार्बन पे आ गए, right? And now this 4 carbon containing compound is changed again, not changed, it still remains a 4 carbon containing compound. Now something is happening over here. Is this ATP coming out or going in?
Going out.
Coming out. तो इसका मतलब ATP इसमें बना है, right? So let me use another color. First ATP molecule made, right? अच्छा फिर आगे चलते हैं. 4 carbon containing compound में अब इस 4 carbon का क्या रिमूव हो रहा है? हाइड्रोजन.
Wait, organ का Miss Fad क्या?
Fad is just like NAD. It's also a hydrogen carrier.
अच्छा, okay. So same cheese.
But it's a bit different in structure. But the dehydrogenation. So how many dehydrogenations up till now? 3. अभी तक तो 3 हो रही हैं, ठीक है. How many decarboxylations up till now? 2.
2.
Yes. Decarboxylation is in black color. हाँ और dehydrogenation blue color में है और ATP को मैंने red color में किया है. फिर उसके बाद एक और dehydrogenation हो रही है.
Yeah.
4. Okay. So in total we have 4 dehydrogenations, 2 decarboxylation and 1 ATP formation. ठीक है. अब आप मुझे बताओ, is this cycle happening twice or once?
Once.
Or twice?
Why would it be happening? Why would it be happening twice?
Because how many molecules of pyruvate did we make?
Oh, there too. हाँ सही है.
ठीक है. So now let's just sum all this up. अब उसको दिमाग में रख के बताओ, how many decarboxylations?
2.
In total, दोनों cycles को मिला के बताओ. How many decarboxylations?
4.
4? 4.
Yeah.
Okay, so 4 decarboxylations. How many dehydrogenations?
8.
8 dehydrogenations. How many ATPs produced?
4. No, sorry, 2.
2. अच्छा, अब इस चीज को गौर से सुनो. This kind of ATP production, I'll repeat once again, this kind of ATP production which is produced when we are breaking down the CH bonds of this 4 carbon containing compound is known as a substrate, because हम substrate use कर रहे हैं और ये substrate यही है, ये compound है, substrate linked phosphorylation. हाँ,
Yeah.
ठीक है. अब मैं थोड़ा सा पीछे जाऊंगी, right? I'll go back and I'll remind you what we were doing in the beginning of the presentation. कल हमने जो किया था, हमने ये किया था कि I told you that the energy for the ATP synthesis can come from chemical potential energy, and the energy for the ATP synthesis can also come from the electrical potential energy. तो अगर मैं कोई chemical use करके ATP बना रही हूँ ना, तो that is what substrate linked phosphorylation. मैं इसको क्यों कह रही हूँ substrate linked phosphorylation? Substrate इसलिए कह रही हूँ कि मैं substrate use कर रही हूँ, और phosphorylation इसलिए कह रही हूँ कि I am using inorganic phosphorus to bind to an organic compound. So by that definition, it is phosphorylation. और मैंने बनाया क्या? ATP बनाया. ठीक है. Let's move forward to see कि क्या हमने यही किया है यहाँ पे. बिल्कुल यही किया है. You'll see that. Look here. I'm now rubbing all this to explain you substrate linked phosphorylation. ये जो आपका ATP बन रहा है ना, इधर इसको देखो. ये जो आपको रिएक्शन दिखाया हुआ है ना, तो ये ATP किस चीज से बनाया? एक substrate से. So the substrate is this compound, and this reaction, although it's written in a very simplified manner, लेकिन असल में ये reaction ये रहा.
Okay.
और ये जो है वो यहाँ से आया है. Hence the name substrate linked phosphorylation. Get it?
Yeah, I get it.
ठीक है. अच्छा, अब पीछे चलते हैं, थोड़ा सा ग्लाइकोलिसिस पे चलते हैं. उसमें भी there is something similar happening. Let me just show you this. Yes. Was there any ATP being formed in glycolysis? बन रहा था ना ATP? कितने बन रहे थे टोटल?
Yeah. दो
4 बन रहे थे ना. 4 मतलब टोटल तो वो नेट तो 2, लेकिन 2 दफा बन रहे थे ना, राइट? अच्छा, ना अगेन ये भी इन्होंने बड़े मजे से आपको बता दिया कि एटीपी बन रहे हैं, बट दिस इज आल्सो सबस्ट्रेट लिंक फॉस्फोराइलेशन. व्हाई? बिकॉज व्हाट्स द सबस्ट्रेट ओवर हियर? दिस इज अ सबस्ट्रेट. दिस इज जो आपका इंटरमीडिएट. हाँ, ये भी सबस्ट्रेट है, ये भी सबस्ट्रेट है. और अगर मुझे कोई कहे कि यहाँ पे एक्चुअली क्या हो रहा है, तो यहाँ पे एक्चुअली ये हो रहा है. ये फौरन से इन्होंने जंप करके दिखा दिया कि ये बन रहा है, एक्चुअली ये हो रहा है. सबस्ट्रेट लिंक्ड phosphorylation. Same thing. Got it?
Got it.
Same thing over here as well. इसी अब आपको जब आपको substrate link phosphorylation समझ आ जाएगा ना, तो जब मैं आपको oxidative phosphorylation कराऊंगी कहाँ पे, which happens on the crystal of the mitochondrion, जो electrons की motive force से phosphorylation होती, तो then you will be able to understand कि अच्छा इस phosphorylation में और oxidative phosphorylation, what's the difference, right? Okay, so let's अच्छा, think about it. ये सवाल आपके जहन में नहीं आया? मुझसे तो बहुत स्टूडेंट्स पूछते हैं कि बस व्हाट्स द पॉइंट ऑफ गोइंग राउंड एंड राउंड एंड राउंड. मतलब we started with 2, we are adding 2 to 4, and then we're getting 6, and then again 6 से फिर 4, फिर 2, ये गोल गोल का. What's the purpose? What's the purpose? Think about it.
The purpose.
In other words, I can say, what are we gaining from the Krebs cycle? Are we mad to keep going in this cycle? to keep the molecules just being converted from one form to another?
I don't know, miss.
Okay, so this is the logic of the Krebs cycle. Go through it.
The release of hydrogen, which can be used in oxidative phosphorylation to make energy. So no direct involvement of molecular oxygen, but will eventually stop. That's all hydrogen carriers will be loaded already. इसका मतलब क्या है?
इसको छोड़ दो, ये नहीं समझ आएगा, बिल्कुल नहीं समझ आएगा, because we haven't done oxidative phosphorylation. Basically, this is your concern right now. The reason why we want the Krebs cycle to happen is we want lots of hydrogens. Let's go back. Let's see how many hydrogens have we collected. कितने collect किए हमने hydrogen? Keeping in mind that we have 2 pyruvate molecules. ठीक है, एक pyruvate नहीं है, 2 हैं. तो कितने collect किए आपने hydrogen? बताओ मुझे.
8.
8. अच्छा, पीछे कितने collect किए यहाँ पे? 9. Right? और glycolysis में भी तो collect किए थे ना? ठीक है, बहुत सारे जमा हो गए अभी. So basically the contribution of the Krebs cycle is that it allows lots of hydrogens to be released from the compounds, different kinds of compounds, so that we can finally, when we reach the stage of oxidative phosphorylation, we can use them to provide energy to make ATP. इसलिए कि ये वही हाइड्रोजन है जो अब स्प्लिट होंगे into protons and electrons. हम जब ये स्प्लिट होंगे, we will be using the energy of those electrons to make ATP. And do you remember मैंने क्या कहा था कि हम जो है ATP केमिकल पोटेंशियल एनर्जी से भी बना सकते हैं, इलेक्ट्रिकल पोटेंशियल से भी बना सकते हैं. तो ज्यादा एटीपी किससे बनता है?
क्रेब्स से नहीं, सॉरी, उससे.
नो,
क्या कहते हैं?
इलेक्ट्रिकल से या केमिकल से?
केमिकल.
नो, read this once again. This is what I told you yesterday.
Oh yes, electrical. Large amount of ATP is made and it's kind of small size.
Right? Okay. So now let's do some balance sheet. This is the balance sheet of the Krebs cycle. अच्छा, the balance sheet which I have made, just look at the top. मैंने क्या heading डाली है? Krebs cycle per 1 glucose molecule. तो per 1 glucose molecule का मतलब कितने pyruvate? How many pyruvates? 2 or 1? 2. ठीक है. So from that perspective, the number of reduced NADs that I got from Krebs cycle are 6. क्योंकि 3 दफा dehydrogenation हुई तो 6. The number of reduced FADs that I got were 2, and the number of ATP molecules that I got were 2, and the number of CO2 that I got were 4. Right?
4.
ठीक है. Okay. अब हम करेंगे. We are doing some calculation of how many ATP molecules we can actually get from all these reduced NADs and FADs. ठीक है. अब होता ये है कि this is a rule कि every single reduced NAD, अब ये rule है, fixed rule है, ठीक है. Every single reduced NAD will give you 3 ATP molecules, and every single reduced FAD will give you 2 ATP molecules. ये एक rule है आपको जो नजर आ रहा है स्क्रीन पे, ठीक है. Glycolysis में how many reduced NADs did we make? 2. 2 बनाए थे हमने glycolysis में.
Yep.
ठीक है. अच्छा, तो अगर एक NAD 3 ATP देता है तो how many ATPs will I get from 2 NADs? 6. अच्छा, link reaction again gave me 2 NADs.
लिंक का भी सेम है, 2 ऐड है तो अब उससे 6 हो जाएगा. Krebs में
Krebs का
NAD but shouldn't be, but
FAD कम ATP देता है, ठीक है. FAD only gives us 2 ATP, so then this will be 18 plus 4. So how many ATPs altogether?
34. Yeah.
34 ATPs, ठीक है. Just remember this number, because why am I actually going मेकिंग यू गो थ्रू दिस एक्सरसाइजेस कि जब हम अभी ऑक्सीडेटिव फॉस्फोराइलेशन करेंगे तो ये नंबर हमें काम आएगा. वी नीड टू रिमेंबर दिस नंबर, राइट? ओके, नाउ लेट्स मूव फॉरवर्ड टू. हाँ हाँ, अच्छा, ये जो मैं 34 नंबर निकाला है ना, what have I done? I have kept this number under the heading of oxidative phosphorylation, and I have kept the number 4 ATPs under the heading of substrate linked phosphorylation. इसका मतलब क्या है? इसका मतलब ये है कि which of all those ATPs जो मैंने substrate को तोड़ के बनाए थे ना, when I was breaking the CH bonds and I was ending up with ATPs, those are purely those ATPs which come under the heading of substrate linked phosphorylation. All the ATPs that I got from reduced NAD or reduced FAD are considered to be ATPs which come from oxidative phosphorylation. इसको हम oxidative इसलिए कहते हैं कि अभी आगे जाके oxidation reduction reactions होने वाले हैं बहुत सारे. अभी हम क्या करेंगे कि इन NADs और FADs को use करेंगे for oxidation and reduction reactions on the cristae of the mitochondria. तो substrate link से 4 बने. कैसे बने 4? I'll just show you कैसे 4 बने. देखो, एक substrate link से हमें मिले 2 यहाँ से. We got, what was the net ATP that we got from glycolysis? 2. 2. What was the net ATP that we got from Krebs cycle? 2.
2.
Krebs cycle से भी तो 2 मिले ना? ठीक है, ये रहे 2. मैं इस ATP की बात कर रही हूँ, इस वाले ATP. 2. So 2 plus 2 4. ठीक है, 4 ATP from substrate link phosphorylation, 34 ATP from oxidative phosphorylation. I hope I am making sense to you. समझ आ रही है बात या नहीं समझ आ रही? ये वाली बात
नहीं सर, सॉरी नहीं हो सकेगा.
स्क्रीन शेयरिंग में थोड़ा टाइम. यू गेट इट ना? इस पे काफी बच्चे कंफ्यूज होते हैं. सो 4 ATPs from substrate link phosphorylation and 34 ATP. Now you see, अभी पीछे मैंने स्लाइड पे क्या बताया था कि ज्यादा ATP कहाँ से मिल रहे हैं? केमिकल पोटेंशियल से या इलेक्ट्रिकल पोटेंशियल से?
इलेक्ट्रिकल.
ये जो है ना, this is electrical potential energy and this one is chemical potential energy. Anything, जब भी आपको ये नाम नजर आता है ना, substrate linked phosphorylation, इसका मतलब ही ये है कि ये chemical potential energy है. And chemical potential energy will always give you less ATPs. Electrical potential energy will always give you more ATPs. ठीक है, अच्छा ये हो गया, ये भी हमने कर लिया. We did all this. You can also remember this. One of the students asked me, so I gave him the answer. He was asking me, Miss, what is going to end up, like what's the fate of all those carbon dioxides which we have released by decarboxylation? So, वही तो वो carbon dioxide है. Whenever we write this equation, हम ऐसे comprehensive equation लिखते हैं ना, summarize equation लिखते हैं cellular respiration की. So don't we write 6 carbon dioxide molecules? Balance जब करते हैं equation.
Yeah.
So ये कहाँ से आए? These carbon dioxides have formed because at so many steps decarboxylation हो रही थी. तो what is the ending of this carbon dioxide? Ending ये है कि जैसे ही ये बनेगा, it will be removed out of the cell as an excretory product. Carbon dioxide cell से बाहर निकलता है, finally फिर blood वहाँ जाता है, lungs में जाता है, and we breathe it out. ठीक है? So you will say, examiner can ask you, suggest where does the carbon dioxide go, which was released by decarboxylation? तो आप कहोगे कि it will diffuse out of the cell and finally be exhaled out of the body. ठीक है? अच्छा.
सही है.
हाँ, ये तो हम करेंगे बाद में. Okay. Oxidative phosphorylation. I won't be able to finish it, but जितना हम कर सकते हैं हम कर लेते हैं. What you can see in front of you is a mitochondrial membrane. This is not the cell membrane. ठीक है, ये mitochondrial membrane है. और हम कहाँ हैं? हम माइटोकॉन्ड्रियन के अंदर हैं. अच्छा, this is the inner membrane. What's the other name of the inner membrane?
Matrix.
Inner membrane का दूसरा नाम क्या है?
Matrix.
This is the
Inner membrane में क्या
mitochondria.
होता है? एक second. Like wait.
ये मैं बना रही हूँ inner membrane. You can see I'm making it. What am I making actually? Outer membrane. Inner membrane. Christy.
Yeah.
ठीक है. अच्छा, this is the matrix. ये. ठीक है. सही है. अच्छा, so imagine that we have zoomed into the inner membrane. ये सारा कुछ inner membrane ही है. So this is your inner membrane, just like any other membrane. It's a phospholipid bilayer. ठीक है. याद है जब मैंने ये कराया था? Let's go back. This diagram. What is studied on the inner membrane? What do you find on the inner membrane?
ATP synthase का stopped particle.
And what else? What is this?
Electron transport chain.
These are carrier molecules,
so
right?
Carrier proteins, right? Yeah.
ठीक है, ठीक है. अब इसको याद रखना जरूरी है, otherwise you will not understand. Now, same inner membrane आपको नजर आ रही है. Look at the stalk particle. ये देखो. ठीक. As I told you, the stalk particle has two parts. One is this part, which functions as a channel protein, and the other is this goal wala part, which functions as the enzyme ATP synthase. ठीक है. तो ये function करेगा as a channel protein, ये function करेगा as an enzyme. और क्या चीज नजर आ रही है माइटोकॉन्ड्रियल मेम्ब्रेन पे? What are these? याद है वो red dots क्या थे?
Yeah, electron transport chain.
Electron. So what is an electron transport chain? It's nothing but it's a chain of transport molecules. It's a chain of carrier molecules. Their name indicates that they are transporting electrons. ठीक है. बड़ा ओवरव्यू करके और सिंप्लीफाई करके बता रही हूँ, डिटेल तो अभी बिल्कुल नहीं बताऊंगी. कि you can very easily see कि सारे NADs and सारे FADs who were sort of carry कर करके load कर करके ला रहे थे अपने साथ क्या hydrogens, वो आके and they are docking onto these electron transport chains, and what are they doing? All hydrogens are splitting into protons and electrons. Do you see?
Yeah.
सब electrons में split हो रहे हैं और आपको नजर आ रहा है कि electrons jump कर रहे हैं from one electron transport protein to another.
Chain to one another. Yeah.
हाँ, इनके jumping से जो है ना आती है energy. The jumping actually gives energy to facilitate the formation of ATP with the help of the enzyme and with the help of the channel protein. I'm going actually very basic because abhi time nahi hai, because there's just 2 minutes left.
Yeah.
And I also have to hurry for Jumma and all. So this is how it is. Okay? We are using the energy of electrons to make ATP. Since this is a jumping of electrons from one protein to another, so for example, हम क्या कहते हैं? Removal of electrons क्या होता है अब्दुल्ला? Oxidation या reduction?
Wait, H plus you're saying add हो रहा है ना?
नहीं, इलेक्ट्रॉन्स की बात करो. आप जब केमिस्ट्री में ऑक्सीडेशन रिडक्शन पढ़ते हो तो ऑक्सीडेशन में इलेक्ट्रॉन्स ऐड होते हैं, रिडक्शन है ना. अब मुझे बताओ, जैसे ये जो प्रोटीन है ना, दिस प्रोटीन, इट गेट्स इलेक्ट्रॉन, इट्स एक्सेप्टिंग इलेक्ट्रॉन. सो इट इज इट ऑक्सीडाइज्ड और रिड्यूस्ड? एक्सेप्ट कर रहा है.
It's wait, reduced.
अच्छा, ये एक्सेप्ट कर रहा है. अब ये डोनेट कर रहा है. अब क्या हो गया इस प्रोटीन को? इज इट ऑक्सीडाइज्ड?
Yeah, no, it's reduced, no?
देखो, what I'm trying to say कि हो क्या रहा है कि electrons are accepted, let's say ये पहला प्रोटीन था, इसने इलेक्ट्रॉन एक्सेप्ट किया, जब ये एक्सेप्ट करेगा तो ये थोड़ी देर बाद डोनेट कर देगा, ठीक है? तो
so that means that protein is oxidized, the other one's produced.
हाँ, exactly. This is what I'm trying to say. So this means
Yeah, no, I get that.
This means that the jumping of electrons along these electron transport carrier molecules is nothing but a series of redox reactions.
Yeah.
ठीक है? ये इसको हम कहते हैं repetitive oxidation reduction reaction. इसकी वजह से एनर्जी आती है, उसी एनर्जी से ये ATP बनता है, and this kind of ATP synthesis or this kind of phosphorylation is what? Oxidative phosphorylation. और यही है वो oxidative phosphorylation जो आपको 34 ATPs दे रहा है. This is not substrate link phosphorylation, right?
Yeah. Substrate was.
So with that, हाँ, substrate दूसरा था, ये 34. So with that we end today's lesson, and inshallah we'll be continuing tomorrow, right? So see you then.
Yeah.
Allah
I'll see you
Hafiz.
tomorrow. So tomorrow you said 12:30?